Hello, my name is Rodrigo, and in this video I'll be talking about high content screening data. We'll start with talking about how we go from images to numbers. The first step is image segmentation, where we identify individual cells within an image. This usually starts with detecting nuclei with something like a DAPI stain, and then expanding outward to define the full cell boundaries. In the CellProfiler example shown here, you can see how this works in practice. Cells are identified and outlined, allowing us to treat each one as an individual unit of measurement. Once cells are segmented, we extract quantitative features from each one. These can include things like intensity, texture, shape, and relationships between channels, such as colocalization. At this stage, it's important to be careful about how data is aggregated. Typically, we measure hundreds of individual cells across multiple images within a well. But for downstream analyses, these measurements are often summarized at the well level. There are different ways to do this, though. For example, taking the mean of all cells, summing values, or aggregating them in more complex ways. And these choices can affect the results. There's also nuance in terms of what's being aggregated, since some measurements originate at the pixel level and others at the cell level. Which one you choose will depend on exactly the biology behind what you're studying. Even something as simple as shape can be quantified in multiple different ways, such as form factor, perimeter, area, longest axis, each capturing a slightly different aspect of the phenotype. So by the end of the process, each cell or each well is represented not just by images, but by a set of quantitative features that describe cellular phenotypes in detail. Once we have the numerical data, there are several important challenges to be aware of. First, the data is extremely high dimensional. We're going from hundreds or thousands of images, each made up of many, many pixels across multiple channels, to cell level or well level measurement that can include hundreds or even thousands of features. Especially in multiplexed assays, this quickly becomes too complex for any kind of manual analysis. Then, many of these features are also highly correlated. This is particularly common in the high content screening platforms that automate feature extraction, where multiple features might be slight variations of one another. Like we mentioned the different ways of aggregating intensity, such as mean versus sum, or other related measurements capturing similar properties. In the heat map shown here in the top right, you can see blocks of highly correlated features, like the squares in the top left and center. These reflect groups of measurements that are capturing very similar information. So while this redundancy can be useful, it also needs to be handled carefully during analysis. On top of that, imaging data is inherently noisy. Variability can come from differences in background intensity, illumination across the plate, or even subtle differences in imaging conditions. Because of this, it's often necessary to normalize data relative to controls within each plate to make measurements more comparable. Finally, the data can contain technical artifacts arising from issues like hardware imperfections, staining variability, or segmentation errors. In the example images shown here, you can see how these artifacts can distort measurements if they're not properly identified or accounted for. And all of these challenges are important to keep in mind as we move forward towards extracting meaningful biological insights from the data. Keeping that in mind, to get to biological insight, we typically go through a few key steps. First, we normalize the data and correct for batch effects so that measurements are comparable across plates and experiments. As I mentioned earlier, this is where per-plate controls become extremely important. They help account for noise as well as positional biases that can occur across a plate. At the same time, it's worth noting the increasing use of deep learning approaches in this space. These models can learn what phenotypes of interest look like directly from images. These can bypass traditional feature extraction and even help mitigate batch effects. For example, in the image shown here, a model has learned to identify binucleated cells and then detect micronuclei within these binucleated cells, capturing a fairly complex phenotype in a more automated way. In direct analyses, though, the next step is often to reduce dimensionality. This can be done using methods like PCA, which we use to more transiently compress the data into a smaller number of components that capture the main sources of variation, or through feature selection approaches that retain only the most informative measurements. From there, we can start identifying meaningful patterns. For example, we might visualize cells in a low-dimensional space, like in this UMAP in the top right, where similar phenotypes cluster together. We can then examine those clusters to understand and label different cellular states or phenotypes that show up after perturbation. We can also define phenotypes more directly. For instance, we might threshold a feature like average GFP intensity in wells to separate these wells into different categories, and then use another feature like average cell size to rank or prioritize candidates, as shown in the figure here. You can imagine extending this idea across multiple features to refine candidate selection and filter out confounding phenotypes to then validate hits experimentally. Thank you for watching, and thank you to the University of Arizona's Data Science Institute, the Colleges of Science and Information Science, and the University Libraries for making this video possible.